I'll first ask Carol Bult, who's a professor at the Jackson Laboratory, to give us a quick update on activities at JAX.
Great. Thank you, Lee. And thanks for the opportunity to give you an update on some of the research happening at the Cancer Center, and also to everyone from SWOG and Hope Foundation for everything you've done to sort of integrate us into this community. We really do appreciate that. So the Jackson Laboratory is an NCI-designated cancer center, basic science-focused cancer center. We have 3 campuses, one in Maine, Connecticut, and here in California in Sacramento. Our Cancer Center program, we have a single program, Genetic Models for Precision Cancer Medicine, and within that we have 3 focus areas, focusing on how cancers begin, how the properties of cancer cell robustness, resistance arise, and we approach those questions through our core expertise in mouse models of human biology and disease, genetics and genomics, and computational biology. So today I have 10 minutes, so I'm going to go really fast. I'm going to give you an update in 3 programs. I only have time for a brief update in each of these areas. Our patient-derived xenograft program, our emerging clinical genomics program, and some really cool science by a colleague on novel therapeutics that he's calling genetic chemotherapy. So most of you, I'm sure, are familiar with the concept of patient-derived xenografts already. We have a long-standing program in this. This is where tumor material or cells from humans are implanted in immune-compromised mice. Tumors are allowed to expand and then taken out and put into more mice to expand that, and then banked, cryoviably frozen. And then at any time, those fragments can be taken out of the bank and grafted into additional mice, and you can expand to get a large cohort of tumor-bearing mice to put on particular drug studies. So we have currently over 300 established PDX models in about 20 different cancer types. And one of the reasons for building this resource is to be able to use these to model drug response either in an individual patient, so you can see you'd have cohorts of mice derived from a single tumor. You can then treat these with monotherapies or combination therapies and look at the response of the tumor volume. But the power of what we've built with this bank is that you can create what we call panels. So you can take from the freezer groups of tumors that share some biological property that you're interested in, and then do sort of a mini virtual clinical trial with this same approach. So we've been working on these kinds of studies with a number of partners, many of whom are SWOG partners at UC Davis, MD Anderson, and others. And the focus for at least 2014 is really to credential this platform as a valid clinical platform. And what that means is taking the tumors in these engrafted mice and subjecting them to standard of care therapy, and making sure that the tumor-bearing mice have a response that mirrors what one would see in the patient. So it's really important, as Lee emphasized, if we're going to build on this, we make sure the science is sound. And so a lot of the efforts this year are going to be around credentialing the models that we have in the PDX resource. The other aspect of this program that I think is really important is the comprehensive genomics that we're performing on all of the models. Not just somatic mutation, but whole genome-wide gene expression, copy number variation. We've got some pilots on whole, that should be whole genome studies, and integrating that information then with the drug efficacy studies in the mouse and the genomics, and really building a platform that combines the excellent clinical annotation we have for all these patient samples with the drug response, with the genomics. This is an unprecedented sort of platform from our perspective for driving forward translational types of research. So our priorities, as I said, we continue to build this resource, characterize and credential the models in the resource, and build that predictive platform by integrating all of this information. The second update I wanted to give you is on the clinical genomics at our Connecticut campus. So we have a relatively new facility in Farmington, Connecticut, the Jack's Genomic Medicine Institute, shown artistic version here, but it is real, and it is coming online October of this year. They're really doing a great job at keeping on that schedule. One of the aspects of this new campus is we're launching a molecular diagnostic service with our Jack's clinical cancer panel. This is a 358 gene panel currently, which covers a lot of the very common targetable genes related to cancer. We achieved CLIA certification for this service last year and are in the process of CAP certification. And I want to emphasize that one of the things that we're very excited about for this panel and we think is unique is, in addition to the usual suspects that one would expect on a cancer clinical panel, we also have a lot of genes on this panel that are there because of research interest around emerging therapeutics. Many of the pathways we've heard about today have representation on this panel. And it provides us an opportunity to work collaboratively with investigators to constantly reevaluate and re-implement this panel. So our plan is to reevaluate the panel every 6 months or so. And of course, to go along with this, we have a well-integrated informatics platform to call the variants and interpret them. So our plan for this year is to complete CAP certification, launch the service this summer, and to start reevaluating the panel based on the data we have so far. So the final update I would like to give is on this concept that we're calling genetic chemotherapy. So this is an emerging concept that's coming out of one of the laboratories at Jack's, the laboratory of Kevin Mills. And essentially, the concept is that genetic instability is the root of tumor evolution, progression, and resistance, and that genetic instability is in fact targetable. So this is from Kevin's research, and the basic idea here is that RAD51, which is essential to DNA repair, there are actually different RAD51 involved reactions, including cancer-specific RAD51 reaction. And Kevin has, through drug screening, has found a compound that seems to specifically target the tumor reaction for RAD51 for DNA repair, and especially in cells that express a genome instability marker. So what you're showing here is the work he's done on this compound C15231A that shows that it is in fact a potent RAD51 inhibitor, and that it's highly selected for these cells that express this genome instability marker, AID plus cells. And the work he's done so far is sort of demonstrating this with AID plus lymphomas in vitro, and you can see the very clear pattern here of the potency of this with AID plus versus AID minus cancer cells. So the way forward in this research is Kevin has work in progress to do a phase 1 trial that is going to focus on B-cell lymphomas. So very, very brief. I know that was a lot in a very short amount of time. I do want to emphasize that our cancer center has an amazing array of researchers working on a lot of transdisciplinary, highly integrative research. And many of those research programs have translational relevance that we are trying to push forward. I don't have time to talk about it now, but I am around for the rest of the meeting. I have colleagues here who I'm going to introduce in a minute, and any of us would be happy to discuss further the other kinds of research that are going on at the center. So thanks very much again to the Hope Foundation, to the partners here that we're already working with, and with my remaining 24 seconds, I'm wondering if my colleagues from Jack's would stand up so that people can know who to talk to. So Ed Liu, of course, I don't have to introduce Ed Liu, our director, is here, just flew in from Geneva. Barbara Tennant, if you can raise your hand. Barbara is our Associate Director for Research Administration, knows everything about the Cancer Center. What are all our secrets? Yijun, I didn't even know was going to be here. I'm very happy to see him. He's our Director for Genomic Technologies from our Connecticut campus. Joan Malcolm is heading up our cancer clinical work in Connecticut. I can tell you everything about the cancer panel. Susie Earhart. Susie and I work very closely on the whole PDX Avatar program. Wouldn't be anywhere without Susie. Many of you already know her and work with her. And Jim Keck, who is our new Senior Director for in vivo pharmacology and clinical lab services here in California at the Sacramento campus. So all of those folks are here, and any of us would be willing to talk to you more about any of these projects. So Kevin, is Kevin here? There's Kevin. Kevin Mills, genetic chemotherapy. You didn't stand up. There. Kevin Mills is also here. So if you're interested in this genetic chemotherapy work, please do seek Kevin out at one of the breaks. So with that, I'll stop, and thank you very much.